Hi guys, I'm going to record a little intro to muscle physiology lecture for you guys. This will primarily be preparing you for the real physiology by letting you know which parts of the anatomy you need to review. A couple things before I move on, and that is that you have a quiz that is due, I think, tomorrow over endocrine, and then I posted another quiz that is endocrine and muscle that I believe I may do a week from tomorrow, a week from Tuesday. That's getting you ready for your test, and your test is scheduled for a week from this Wednesday. So that should be, let's see. Yeah, it's scheduled for the 11th. That's the date of your test two. Test two will cover endocrine, which you've been working on for ages, and muscle physiology. Muscle physiology is not super hard as long as you remember the anatomy, which is what I'm going to try to do for you guys right now. One other little thing before we get started. I was going to show you this today in class, but I forgot. So I thought you guys might be interested in this from the BBC. This was comparing as of 2016. It's not actually the most current, but it's probably even gotten worse from here. The out of pocket for monthly diabetes costs, including insulin and other diabetic supplies, comparing Italy, the UK, Japan, India, and the US. Thought you guys might think that's interesting. Okay, so let's talk about what you are going to need in order to understand muscle physiology. First off, make sure that you are really comfortable with all of the sarco and myo terms that are necessary for understanding the basics of muscle structure, because we have to use those in order to understand muscle function. So all of these you're going to review on your own. Interactive physiology is fantastic for muscular system anatomy review. Here's the specific pages. And then I will start with Roman numeral 2, which is innervation and blood supply to skeletal muscle. This whole set of notes is really about skeletal muscle, but what we use to learn about skeletal muscle would be really useful for us when we get to cardiac muscle physiology, because cardiac muscle physiology is similar to skeletal muscle. It's similar enough that you can just learn sort of the exceptions. Okay, so a couple things about skeletal muscle. First thing is skeletal muscle is really, really vascular. It's very vascular tissue. So this is a histology slide that is stained both to show the skeletal muscle fibers and the capillaries that are snuggling around each one of them. So this is super vascular tissue. It shouldn't surprise you because, of course, it has tremendous oxygen and nutrient needs. So part of the reason that, for instance, a steak, which is a cross-section of cow muscle, is so red is because of how vascular the tissue is. This is another figure of not staining the actual skeletal muscle fibers, just the capillaries around them. So very, very vascular. Second, skeletal muscle is highly innervated. What do I mean by innervated? I mean that every single skeletal muscle fiber has to have a direct synaptic connection with the nervous system. So the nervous system has to tell every single skeletal muscle fiber what to do. That is different than cardiac muscle because, of course, cardiac muscle not only can spontaneously generate contractions, but the cells can also talk to one another. But with skeletal muscle, what you are going to need is, for instance, here is a synaptic terminal going to one cell, and here's another one going to another fiber, and then there's others. So what I want to tell you is if this one, this axon is damaged, this fiber is paralyzed unless and until reinnervation occurs, which sometimes occurs and sometimes doesn't. So highly, highly innervated. The site at which the somatic motor neuron communicates with the skeletal muscle fiber is called the motor end plate. And you see one there, you see one there, you see one there, you see one there. What it actually is going to look like is sort of like this. So here's where the axon terminal or synaptic knob or whatever you want to call it actually comes right up next to the cell membrane right here of the skeletal muscle fiber. and this is called the motor end plate. The motor end plate is highly curved because it gives more room for receptors. And the more receptors it can have, the more it can respond to the neurotransmitter that's going to be used. So this is a single motor neuron making multiple connections on this one fiber. But if I cut that motor neuron, this fiber is paralyzed. So the neuromuscular junction, which I'm going to abbreviate NMJ for a while, is a synapse between a neuron and a muscle fiber. And a single skeletal muscle fiber will only contract under normal physiological conditions when stimulated by a somatic motor neuron. Hopefully you guys remember that somatic is sort of the voluntary division of the efferent nervous system that controls skeletal muscle. And so what would happen, and we'll get into the details of this in a little bit, what would happen is action potential goes down here, somatic motor neuron, neurotransmitter release occurs here, and then that is what is going to precede the action potential that's going to occur. So we'll get into this in more detail when I see you guys on Wednesday, right after we finish the endocrine stuff. Okay, so let's go into the histology of skeletal muscle, stuff that you guys have learned before, but you need to really be able to use the terms comfortably on Wednesday and then on the following Monday. So first off, this is a skeletal muscle fiber. It's the same as saying that this is a skeletal muscle cell. And so it has characteristics that all cells do, but it doesn't look anything like the cells that you, for instance, studied in 1610. And unfortunately, somebody changed all the names of the parts, but it's still similar to cells that you've looked at before, plus some really notable differences. First thing is it's going to be a eukaryotic cell. Since it's a eukaryotic cell, don't be surprised that it has nuclei. So skeletal muscle fibers, you probably remember from anatomy, are multinucleated. This one's only showing one, but a nucleus could be here and here, and the nuclei are peripherally located, meaning they tend to get pushed up against the cell membrane because the middle of the cell is really kind of full, so there's not much room for them. And then the plasma membrane on a skeletal muscle fiber, the plasma membrane here, is called the sarcolemma. And the thing that's important about the sarcolemma is it is basically a phospholipid bilayer with embedded proteins, just like any other cell membrane. But please note that what happens with the sarcolemma is that it's actually going to tunnel deep into the cell. So these little pores that you're seeing right here are actually going to lead into the cell. And they are also made of a cell membrane. And what can happen is if I get an action potential going on this cell, it doesn't stay superficial because it can actually follow the T tubules or transverse tubules down into the center of the cell. So the action potential would run along the surface and keep going on the surface, but it would also go deep into the cell. That will be important for understanding the physiology. So the T tubules are called invaginations, as in vagina, as in tunnel. Invaginations of the plasma membrane. Okay, and then inside the skeletal muscle fiber are going to be a bunch of stuff that's going to be important. First off, since the tissue is so incredibly energetically expensive, tends to have a whole bunch of mitochondria represented here in pink. And the mitochondria and the other organelles are in what would be the intracellular fluid or cytoplasm, but here they changed it and they called it sarcoplasm. So sarcoplasm is just the cytoplasm or intracellular fluid of a muscle fiber. Okay, so multiple nuclei in the sarcoplasm, bunch of mitochondria in the sarcoplasm, and then two other things that are going to be important to you. And the two other things are glycogen. So although it's not showing here, I want you to know that inside the sarcoplasm here is where you would store glycogen. And you guys know what glycogen is. Glycogen is stored glucose. So I'm starting to see a theme here that I've got stored glucose and I've got mitochondria. Okay. And then one more thing, again, not drawn on the picture, but important. Inside the sarcoplasm of a skeletal muscle fiber, there is also a protein called myoglobin. And myoglobin is similar to hemoglobin. And hemoglobin, for instance, binds oxygen and carries it around in the bloodstream. Myoglobin binds to and stores small amounts of oxygen inside a skeletal muscle fiber. So to reiterate, what we have is oxygen stored on myoglobin, glycogen storing glucose, and then boatloads of mitochondria. What happens, therefore, is that skeletal muscle fibers always have the ability to do a little cellular respiration because they've stored the things that are necessary to do so. So they're kind of your emergency response tissue. And even if you haven't eaten for 72 hours, they still need to be able to get you out of the way of a bus or a saber-toothed tiger. Okay. Then the other two things that are inside there that are important to us are going to be these long cables. What this is a big long bundle of proteins. Think kind of like a coaxial cable. This is called a myofibril, and the myofibril is attached at the end of the cell to the cytoskeleton. And this thing right here is the contractile portion of the muscle. So when the myofibrils contract and they're attached to the cytoskeleton, they are actually able to shorten the length of the entire muscle fiber. So it kind of looks like a little Vienna sausage. It's a myofibril. Wrapping around the myofibrils, this kind of blue-gray color that you're seeing right there is what we call the sarcoplasmic reticulum. And the sarcoplasmic reticulum is similar to the smooth ER, smooth endoplasmic reticulum in other cells, but it has a special function. In the lateral sacs, right here by the T tubules, the lateral sacs of the SR or sarcoplasmic reticulum, you store calcium ions. Okay, so keep in mind and please note how closely associated the lateral sacs are with both the T tubule and also with the myofibril itself, and you store calcium ions right in here. Okay, so the myofibrils are rod-shaped and they're submicroscopic. They're 1 to 2 micrometers in diameter, and they run throughout the long axis of the fibers. So what I want to do now is break down this myofibril so that you can see how the myofibril is related to contraction. So if you look here, and you've done this before in anatomy, I just want to make sure you know what you're talking about here. So this jagged line is called a Z-line or Z-disc right here. So one jagged line to the other one produces something, or a repeating pattern called a sarcomere. So here's 1 sarcomere, 2, 3, a part of 4. So what I want to do is I want to blow this up so that I can look at the sarcomeres in detail. So again, here's a sarcomere, Z-line to Z-line. The anatomy book called it a Z-disc. It's the same thing as a Z-disc. So here's the sarcomere, one right here, and you can see it here as well. Here's another one. Okay, and part of a third one. Now the sarcomere is a 3-dimensional structure, but we're going to simplify it and look at it 2D so we can start to get an understanding of how it's related to muscle contraction. So if you look at the myofibril or the sarcomere in detail, what we will see is, of course, that it's primarily composed of 2 proteins, and these 2 proteins are this orange protein that you see in the middle with the things sticking up. That's called the myosin thick filament, okay, right here. And this long skinny protein that's attached to the Z lines, and that's called the actin thin filament. So thin filaments and thick filaments. And that is primarily, but not exclusively, what makes up the whole myofibril is this pattern of thin filaments and thick filaments. So we will see a little bit later that the thin filament is also going to have 2 other proteins wrapping around it. And I'll show you guys those on Wednesday, but they are called troponin and tropomyosin, and they wrap around the thin filament. Okay, then the thick filament has these heads sticking off of it. So these are called myosin heads. And what they do is they will bind to, when given the opportunity, these attractive sites on the actin called the actin active sites. When they form an attachment between the myosin and the actin, it's called a cross bridge, and will lead to cross bridges next time. So when myosin is given the opportunity, it will form a chemical attachment to the actin active site. So what happens is the arrangement of this actin and myosin will give that characteristic striations to the skeletal muscle fiber. So if I dyed a skeletal muscle fiber so that proteins dye darker. Wherever I would have thick or dense proteins, it would be darker. And wherever I would have less dense or fewer proteins, it would be lighter. So what I want you to see is this. So this thick filament is thicker and denser. And what that's going to do is that's going to correlate to what we call the A band. The A band as in DARK. It's not what it stands for, but that will help you. And then in the middle of the A band, there's going to be, it's not going to be quite as dark because I don't have actin and myosin overlapping. The middle portion right here is going to be the H zone. And then wherever I don't have that thick protein at all, it's going to be the I band. So the I band is going to correspond to from right here to right here. This is the I band. And if we go back to look at skeletal muscle in detail, we will see that what we get is this alternating pattern of dark, light, dark, light, dark, light. And what you're really seeing is dark is A band, mostly myosin. Light is A band, mostly actin. Dark, light, dark, light. Okay, so that is sort of the basis of this sarcomere pattern, the A band, the I band, and the H zone. So let's just make sure that you guys understand this before we try to contract it. So the ends of the single sarcomere are going to be the Z lines, also called Z discs. The actin or thin filaments attach to the Z lines, but the myosin does not. The myosin is floating in the middle. It's stabilized by this little M-line protein that doesn't really ever move, so it doesn't matter that much. And so the myosin is in the middle. So here's your sarcomere banding pattern. Now, the only other thing that's kind of interesting, and it's shown on some figures and not others, is that there's this little springy protein right here called titin. And titin, when the cross bridges let go, that springy titin will allow this to relax back to its original position, but it doesn't really change very much about the physiology. Okay, so the A bands and the I bands are the visible dark and light striations. And then if we go back, we can say, okay, so what's the relationship between a sarcomere and a myofibril? Well, if a sarcomere here to here is like a boxcar, then the myofibril is the entire train, right? And what we will see is the more myofibrils you have in a cell, the more contractile proteins you have, and the stronger the contraction can be. So what I would like for you guys to do is I would like for you guys to be able to label 1 through 8 on the figure below. And ideally, what I would really love is if you color code it similar colors to what we're dealing with here. So that, for instance, the actin is kind of that purpley blue and the myosin is that orange color. And then you should be able to label the actin, the myosin, the Z line, the H zone, the I band, the A band, and the whole sarcomere. Okay, so what I want to show you is a preview of things to come of how this is going to correlate to muscle contraction. And I'm going to show you with this little video, and then we will be finished for today. I'm going to upload this for you and then send you a Canvas notification that it's here. And this is where I will start on Wednesday after endocrine. I will start right here with Roman numeral 4 to show you the sliding filament theory. Okay, so I'm going to let him talk for a minute.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width.
Okay, so let's look and make sure this corresponds to what you guys said. So this would be a relaxed sarcomere. Here's the H zone, and here is the I band from the end of one myosin to the beginning of the other. So the H zone and the I band would be at maximum width when the thing was relaxed. But then what we are going to do is we are going to slide these filaments past one another, and he's going to introduce you to that.
During contraction, the actin and myosin myofilaments interact. The actins slide toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap, the H zones disappear, and the I band becomes very narrow.
So hopefully that makes sense to you guys. I'm going to turn off the audio and then go one more time so you can see it. So relaxed muscle is very stripy, but contracted muscle After the myosin heads start to form cross bridges, and they basically ratchet the muscle closed, ratchet, ratchet, ratchet. The H zone is decreasing in width, and the the I band is decreasing in width until, for instance, contracted muscle would be way, way darker than relaxed muscle. Okay. What this leads us to is what we call the sliding filament theory. So each of these are called filaments, thin filament and thick filament. And in order to make a sarcomere contract, what happens is the filaments actually slide past one another, right? So really myosin kind of stays in the same position and pulls actin into it, okay? So it's called the sliding filament theory or the sliding filament mechanism of contraction. And so what we will do is we're going to get some more detail about this on Wednesday, specifically how ATP is used. And then we'll back up and we'll talk about, okay, well, how does the nervous system stimulate all of that? So let's look here and see if you guys can tell the difference between a relaxed sarcomere with a wide H zone. This is the electron micrograph of this. And the H zone is right here in the middle of the A band, the H zone. And then a contracted sarcomere in which the H zone almost completely disappears, and look at the width of the I band. So here's the diagram, right? Relaxed, contracted, and here's the photo, relaxed, contracted. Okay, and we will stop there for today. I'm going to upload this and then I will send you guys a Canvas announcement. And on Wednesday, I would like for you guys to have watched this and probably labeled that sarcomere in your notes. And then right after we finish endocrine, we'll get into the actual muscle physiology.